last part of the lecture is going to be unit operations. I don't know whether all of you are now able to view the screen. I've shared the screen. Are you able to see it?
Yes.
Okay.
Yes.
Now, Bashar, who is Bashar? Bashar, mute yourself so that you remove your. How do you listen to me when you are having music on? Anyway, so today we are going to handle basic unit operations, and these operations do work not only in pharmaceutical industries, but even in other types of industries. So we are going to see what are the operations or the processes that we actually do. What's this? Okay, we are going to have an overview of what a unit operation is. The history and background, maybe that one can also help us, just as you know how Napoleon began and all those things. History and background, I usually don't pay much attention to it. But if you feel that is important, then we shall handle. Then we shall know the difference between a unit operation and a unit process. Then how do we classify them and how do we apply them, especially in pharmaceutical industry, in pharmaceutical engineering, pharmaceutical manufacturing. So a unit operation gives idea about the physical. That is a unit operation. Unit operation gives an idea about specific physical operation. That means that there is no change in the physical properties of whatever you are transferring to or you are dealing with. They also give an idea about the different equipment that is used in this unit operation and their design, the material from which it is constructed, and actually how you can operate it. There are some calculations that are involved, various calculations that are involved. Some of them we have already seen them, like in heat flow. We have seen some. We have seen some in food flow, and there are many other things that, many other calculations that are involved. And then in pharmaceutical engineering, a unit operation is a basic step in a process. So that means that a unit operation is a subset of a unit process. So it is a unit, it's a basic step in a process. It involves a physical change. Unit operations involve a physical change or a transformation, such as a chemical transformation, such as separation, crystallization, evaporation, filtration, and some of these are actually unit operations which we shall be able to look at when we in detail in this particular lecture. So the history and background, you'll be able to know how it became, it began, why is it so much emphasized in pharmaceutical engineering, and all those things. So you'll be able to look at that. You can even go and Google some of these things here. Now, what is the difference between a unit process and a unit operation. Now, a unit operation is a physical change, while a unit process is a chemical change. So we have chemical conversions in unit processes like chemical reactions, biochemical processes. There are so many processes involved like nitration, oxidation, you can say you are putting a metal into some medicine. That is also a process. So that is a unit process. While in unit operations, the processes that involve are only physical. They are physical to material, to the material, and this is actually termed as a unit operation. So some of them we have mechanical separations, as we shall be able to see in some of these unit operations in fluid flow. Process heat transfer, that one we have done it. Mass transfer, we haven't done it, but I think we shall be able to do it. Then heat and mass transfer. These ones do occur the same, like you are transferring one mass, but somehow there is change in heat. That one is also a unit operations. So unit operations may be tied with the unit process. They can actually occur, just as we have seen it, simultaneous heat and mass transfer. So you can have a physical change and at the same time you have a chemical change. For example, when heat flows into an endothermic chemical reaction or out of an exothermic reaction. So when you heat flows out, the thing cools down. So if it was a liquid, for example, and then it forms a solid. So there is some process which is both physical in that it has changed from water to solid, but there is also some chemical reactions that may be involved, like say bonding and whatnot. The unit operation may also be distinctly separated from a unit process. So one unit operation may occur, then later you can have a chemical change. For example, if you have flow of liquid, the liquid is moved from one part of an industrial establishment to another. In this case, you are moving the liquid from one point to another. In that case, you can say move a gas, then after that you cool it, and that means you have a liquid. So in here, you have physical change, then later you have a chemical change by, say, cool, it can form solids. You look at an example of crystallization, as we shall be able to look at it. When you seed, then you create more crystals being formed. This is the process which is used in sugar manufacture. You have components that if you seed them, there are many other crystals that appear. So that is both a physical and a chemical change, but they all occur at different times. So the different types of unit operation of what, so that you can be able to understand. Let's look at flow of fluids, which we did, and I think we are not going to waste a lot of time on this because we did it and you have the notes. The Bernoulli theorem, the Bernoulli Reynolds number, Bernoulli theorem, and if you don't have them, then we'll be able to understand them through here. And how actually we measure fluids using manometers or seismometers and all these things. I think you have the notes with you. But maybe what we didn't handle are the types of pumps when we are handling fluids. But if we can briefly discuss this, these are mechanical devices used to increase the pressure energy of a liquid. We use pumps, for example, in filtering. We are transferring a fluid, like say a liquid through very small pores, and we are using a vacuum pump to make it to apply pressure so that it can go through. So that is the pump that we use. The different devices, the different constituents of pumps are given in here. And when you read, you'll be able to understand how actually these particular pumps operate. We have different types of reciprocating pumps. We have rotary pumps and many others. But right now we are going to, I think, deal with three: the rotary pumps, reciprocating pumps, and then the peristaltic pump, which contains silicone rubber tube in a U-shaped against the. So all these ones here are going to be sent on to you. They are pictorial to be sent to you in the notes that I'll be able to send to you. Then heat and mass transfer. These ones you have heard, you've seen how heat is transferred through the mechanisms, and they are well discussed in the notes that you have, and even what we mean by a black body as opposed to a gray body. All these ones here are transferring mass. In transferring solids, there are certain things that also you need to know. For example, belt conveyors, screw conveyors, pneumatic conveyors, and what do they do? These ones are really very well explained in this. And this is actually mass transfer, the conveyors. We have conveyors which can even transfer people from one point to another. These ones usually you meet them at the airport, you meet them you meet them at the airport, in supermarkets, and all those places, the different types of conveyors. Yes. So we have also chain conveyors, bucket conveyors, and all those things. Now, the third look at right now, filtration. Filtration, the suspension of a solid and liquid to be filtered, is known as the slurry. The porous medium used to retain the solid is described as the filter medium. The accumulation of solids on the filter is referred to as the filter cake, while the clear liquid passing through the filter medium is known as the filtrate. So we have four terms. We have the slurry, we have the filter media, we have the filter cake, and the filtrate. Now, I think you've done some filtration. Like, for example, if you use a filter paper, what you have, what the material that you are going to filter is what now I call the slurry. The filter paper is the filter medium, while what remains remains on the filter media, the filter cake. And what goes through the filter paper is now called the filtrate. Now, there are many characteristics that affect filtration. And one, the properties of the and corrosiveness. I think most of you may be knowing the plant okra, yes, okra, the green, it's called lady's finger. When you cut it in small pieces and put water, hot water in there, it forms a very thick liquid. Now, sometimes you may need to filter this. You may not be able to use a filter paper unless maybe you use a pump, a vacuum pump, to force this particular thick liquid through the filter paper. So the density actually depends on the density, the viscosity, and sometimes the corrosiveness, because if you want to say filter the same thing, okra, lady's fingers, the funnel may not be metallic. If it is metallic, then it has to be stainless steel. Sometimes this one may cut, may actually eat up the funnel. So it depends on the corrosiveness, the density, and viscosity. The properties of the solid, for example, particle shape, particle size of the solid. So all these factors here do affect filtration. So you'll be able to get them. And so filtration is the process of separation of solids from fluids by passing the same through porous medium that retain the solids, but allows the fluid to go through. So the fluid that goes through is what we have known as the filtrate. So types of filtration, we have surface filtration, we have deep filtration, and examples are given. Then we have cake filtration, and examples are given. So the mechanism of filtration is given here, very elaborate. We can strain. Through straining, it is similar to sieving. That means the particles of, so that is now sieving. Sieving, you just shake so that the big particles do not go through. The small ones do go through. Solids move with streamline flow and strike the filter medium. So the solid move with, there is a streamline flow, and then they strike the filter medium. Now, obviously, the filter medium will only allow bigger particles to go through. Then the entanglement, then the particles become entangled in mass of fiber due to the small size of particles than the pore size. So you find that the bigger particles may not be able to go through, so they get more entangled. They get stuck to the fiber, so that the smaller particles can go through the pores. Now you also have attractive forces. Solids are retained due to attractive forces between the particle and the filter medium. So those which are not attracted will obviously go through. So those are the types of different methods of filtration. Then we have filter aids. So is a group of inert materials that can be used in pre-treatment. So you want to get your products go through. So what you do with your celery is to treat it with another chemical. You can see that now we are having some chemical transformation. It's a unit process. And then you filter. You have it as what? A unit operation. Then that one aids to filter, so that you can be able to do the filtration as fast as possible. I presume that most of you know what a filter paper is, but obviously we have different types of filter papers. We have those ones which have got bigger bigger, they have big pores, and those which have got smaller pores. So it is always important to know the size of particles that you may need. Now we have the different types of laboratory filter papers. Those ones you'll be able to get them. Then we have the chromatography papers. What their application, the high quality papers of chromatography, they can be, you put them, you spot at the end, you spot at the end of it, and then you run through a solvent, they'll be able to have, you'll be able to have what? You have the chromat separate. That means you can be able to filter so that the solids can get stuck onto the paper and then the liquid can move out. So that is also chromatography paper. Now, electrophoresis and blotting, this is some of the methods which are used, for example, in separating DNA, and these ones you can be able to apply them. There are types of chromatography, which I think we shall be able to deal with when we come to chromatography. Rate of filtration, the mathematics that is involved is actually okay. You'll be able, and it is more detailed with the notes that I'll be sending, which will be attached to whatever you are seeing. Filtration equipment given in here, and some of the diagrams for filtering can be given. The different types of industrial filters. You have the centrifugal filters, you have pressure filters, gravity filters, vacuum filters, and what they actually do. They have been given here. And we have centrifugation as a unit operation, like a centrifuge, which we have in our lab, and they usually separate by centrifugal force according to their size. They can also be separated according to their shape, density, the viscosity of the medium, and even the speed of the rotor. So the theory is well explained here, and even the mathematics which is involved. Then we have the size reduction. Size reduction, most of you have done that. You have made for example, the grains of maize into flour. That is size reduction. And what type of size reduction, what type of mills do you have to use? If you are to move from solid to solid, there's no physical change there, but the operation. So there's no chemical change, but the physical change is there from solid grains to powders. And that is also, and the factors that affect particle size separation, solids, mixing of solids. It is a unit operation in which two or more of the two components which are separate are mixed. So each particle lies as nearly as possible. They get together. So you are trying to get two solids, then you are trying to mix them. Because you are after having homogeneity, you want to increase diffusion and dilution to facilitate dispersion, to ensure stability and uniformity, and to promote chemical reaction. So you do this mixing of solids. Some of these are applied. They are applied in manufacture of tablets. So you add their excipients, and some of these excipients which you do are actually because of some of these objectives that you are able to see. So the mechanism of mixing, For liquid mixing, it requires localized mixing and general movement, while for powder mixing, it is a neutral mixture. You just get some mixture, and then sometimes you can even wrap them in a small cavera and then shake. That could also do some good mixing. There are so many other mechanisms which are involved, and they are easily given, they are well given here. We have liquid mixtures and solid mixtures. We have semi-solid mixers. And after extracting, what we get is process is well given here. Those who have started doing their research, I think, are following this particular process of maceration so that they can be able to get out the. Anyway, so evaporation is one of the unit operations that are also used in the manufacture of drugs. And the factors which you have to consider, put into consideration when you are evaporating, are given in here. We do use a lot of evaporation in our lab, especially when we are trying to dry plant extracts. We evaporate off the water. We have a rotary evaporator in the lab. How do we do that? How do you control the temperatures? How do you increase the surface area of extract of evaporation and all those things? They are really very well explained in that. Crystallization is also another process. It's a unit operation. Crystallization is a chemical solid liquid separation technique in which the mass transfer of a solute from the liquid solution to a pure solid crystalline phase occurs. So we have seen, for example, when you want to separate salt from water, you concentrate it. That means you evaporate off, and then the crystals start forming down because the concentration has reached what we call supersaturation. But you can also increase the crystallization by putting there a seed. That's what we call nucleation. You put there a nuclear, and that one is a spark off to the formation of crystals. And to be very well explained here is what we call crystal growth. And where is it applied in our industries? We will be able to do that. Drying is just unit operation, which is different. When you are doing evaporation, you are trying to dry. But then there are some solids, for example, which may not need to use the heat changes. So what do you do? So you supply some heat, some small heat. How do you control it so that you can be able to dry? But you can also use air. Air, you can pass air through the solid, which is wet, and then the air, the dry air, will absorb the moisture and then remove moisture from whatever wet solid you have. So the types of dryers are given here. Now, how do we apply unit operations in the manufacture of drugs? We use these unit operations in formulation and pre-formulation development. Before a drug can be manufactured in this scale, goes into the actual formulation of the drug. So formulation development must, so the formulation developing scientists must evaluate a compound for uniformity, stability, and many other factors. So during this particular time of evaluation, there are so many unit operations that come into play. So you have to make sure that you use them so that you can be able to come up with solid or semi-solid, immediate or controlled release, tablet or capsule, whatever. There are so many that are involved. Then we have powder blending. Powder blending. This one here is also an application. It's a unit operation which you can be able to apply in the manufacture in the pharmaceutical industry. Milling is also a unit operation. Granulation is a unit operation, and I think we've done that. Then we have extrusion. Hot melt extrusion is also another unit operation that you can be able to do in the process of manufacturing drugs. I want to thank you. And I'm going to send these notes through Clover and Emily.